Good morning. Today's topic is about fish diseases. Look at this slide. You can see this fish is very sick and he needs our help. So what shall we do? We don't know what kind of disease that affects this fish. So there are several fish diseases. We have viral disease, bacterial, parasitic, fungal, nutritional, and disease caused by toxic substance and organic waste. But today's topic will only be focused on viral diseases, especially on nodavirus. Nodavirus comes from the family Nodaviridae. It has two genera, the genus Beta-nodavirus, which targets the fish. That's why it's known as piscine nodavirus. And the type species is the striped jack nervous necrosis virus, or SJNNV, which was reported in 1992. And the other genera is the genus Alpha nodavirus, which target the insect nodavirus. We will focus only the genus Beta nodavirus. Nodavirus is usually non-enveloped, round-shaped, and about 25 to 30 nanometer in diameter. It is icosahedral with a T3 symmetry, and the capsid is composed of 180 proteins. As you can see in the right side of the picture is the virions of nodavirus as shown in electron microscope. What about the genome of nodavirus? The genome of nodavirus is segmented bipartite linear, single-stranded RNA, positive sense. And this consists of two genes, the RNA, which is known as the replicase gene, and the RNA2, which is known as the coat protein gene. So each gene is cut with a 5' and at the end is a 3' with non-polyadenylated RNA. Beta-nodavirus is a causative agent of viral nervous necrosis, VNN, or viral encephalopathy and retinopathy, VER, which infects marine or freshwater fishes and invertebrates. So I have to show to you now the global distribution of viral nervous necrosis, as shown in this slide. The first cases of VNN was reported in Japan in a hatchery-reared Japanese parrotfish, and barramundi in Australia, both in 1990. And it spreads worldwide. That includes the Asian and Southeast Asian countries, and that includes Philippines, which was reported in 2002. And it was also reported in European and Mediterranean countries, and also in the American countries. Why is this very important? Because Beta-nodavirus is one of the important viral pathogens that is reported by the Office of the International Epizootis that infects the cultured marine fish. Usually, this viral nervous necrosis affects about 16 families of cultured marine fish, which consists of about 20 species worldwide, as you can see in this slide. What about the characteristics of viral nervous necrosis? Usually, the target organ of this viral nervous necrosis that infects by the Beta-nodavirus is the central nervous system that includes the brain and the spinal cord and also the retina of the eye of the fish. As you can see in this slide, when Beta-nodavirus infect the fish, it produces baculation and cell necrosis of the tissue of the brain and retina of the eye. And this fish, after 3 days of infection, it usually produces corkscrew swimming behavior, in which it lost its equilibrium in swimming, and then floating behavior usually is produced when VNN occurs. So as you can see in this picture. And here, Beta-nodavirus is shown as a yellow color. or particles as seen in the central nervous system and in the retina in the eye of the nodovirus using the immunofluorescent antibody test. There are 4 genotypes of beta nodovirus. The SJNNV, or the striped jaw nervous necrosis virus, which targets the striped jaw, and usually the optimum growth temperature is between 20 to 25. Next is TPNNV, which targets the tiger pupper, or known as the tiger pupper nervous necrosis virus, which the optimum growth temperature is 20. Next is RGNNV, or the red spotted grouper nervous necrosis virus, which the target source are almost warm water fishes, and the optimum growth temperature is between 25 to 30 degrees Celsius. And the last is the BFNNV, or the barfin flounder nervous necrosis virus, which targets almost all cold water fishes, and the optimum growth temperature is between 15 to 20 degrees Celsius. How does nodovirus is transmitted? It can be transmitted by vertical transmission or by horizontal transmission. As shown in this slide, this is about the vertical transmission of nodovirus. This is from the broodstock to the larvae. So it was already known that nodovirus can be detected in the ovary or the gonad of the broodstock, and it usually pass out together with the egg, but we don't know if this virus usually attaches on the surface or inside the egg. So upon fertilization of the sperm, this infected fertilized egg is hatched into larvae. But within 3 days, after 3 days of infection, almost all these larvae, about 90%, produce mortality. And then 10% that survive usually grows into a juvenile, but we don't know if this juvenile fish carries the virus or not, and it grows into a broodstock and repeat the cycle. But this, in the vertical transmission, can be prevented or can be shut out. How? We can do a selection of virus-free broodstock by using PCR or RT-PCR method. Then, those fertilized infected eggs can be disinfected by using ozonation and also disinfection of the rearing water or rearing facilities. This was already proven to prevent the vertical transmission of broodstock, of the nodovirus from broodstock to the larvae. How about horizontal transmission? We suspected that apparently healthy or diseased wild fish in habits near the aquaculture area are also important source of beta-nodovirus infection that causes BNN. So we can be able to detect beta-nodovirus using the apparently healthy or diseased wild fish near the mariculture area. There are several diagnostic tests of beta-nodovirus. It's either by demonstration of characteristics of lesions in the brains or retina of the fish by using light microscopy, that is by using histopathology and immunohistochemistry. Second is demonstration of virions, viral antigens or viral nucleotides by using electron microscope, RT-PCR and nested PCR, nucleotide sequencing, and phylogenetic tree analysis. Third is detection of specific antibodies by using ELISA and IFA. And the fourth is tissue culture of the virus using SSN1 or E11 cell line. But today, what we are going to discuss only is about the RT-PCR, nested PCR, and nucleotide sequencing and phylogenetic tree analysis. Why did we have to discuss only this tool? Because this tool is the recent tool that was developed and very specific and very sensitive and very rapid to detect beta-nodovirus RNA. So what is the protocol or the procedure on how to do RT-PCR method? So it's very simple. Just extract the RNA from the brain or the other tissues of the fish or invertebrates and run through a thermocycler, gel electrophoresis, and check the PCR product. If it is positive, then we do the nucleotide sequencing. If it is negative, do the second step of PCR method, which is the nested PCR. Then repeat the same protocol until you get the nested PCR product, and if you get a positive band, then do the nucleotide sequencing. But before that, we have to also design a primer specific for RT-PCR and nested PCR. How do we design a primer? Primer usually is designed based on the code protein gene or the RNA2 of the nodovirus. So the RT-PCR primer usually designed from the conserved region to the variable region with a target length of about 524 base pair. And while the nested PCR product, which is the second part of the PCR method, you have to design inside the RT-PCR primer or the RT-PCR product area, which is also between the conserved and variable region. So, in the next slide, we do the nucleotide sequencing. So after you get the PCR product, we do the nucleotide sequencing by removing the conserved region of the PCR product and then get only a small part, which is the variable region, about 177 bases, and run for multiple alignment using the nucleotide sequencing and phylogenetic tree analysis. This is just a software on how to analyze the genotypes of beta-lactovirus. So, if we have done all of these things, then we know that this is already nodovirus. Then we have already confirmed that the sick fish is already caused by viral necrosis, especially by nodovirus. So, since we already know that this is already nodovirus, so we have done already, based on that picture in the first slide, that we have diagnosed it, that this is a viral disease. Viral disease, and especially this is caused by nodovirus. At present, there is no specific management against viral nervous necrosis caused by beta-nodovirus. So, but we have some alternative methods on how to manage or prevent this kind of disease. So in this management, this includes RT-PCR selection of the broodstock or the larvae. RT-PCR. Then if you have selected, use disinfection method. Disinfect. Disinfection method means you have to disinfect the fertilized egg, the infected fertilized egg, or the rearing facilities or the water used to culture the marine fish. And third is vaccination. So we have to vaccinize the broodstock in order to prevent infection. So, is there any question about my topic for today?